Okay, just come stand over here and, oh, sorry, because the sound's just from the computer right now.
Oh, today. Okay. Is this good? Oh.
Yeah, maybe a bit closer to make sure the computer picks up. Okay.
Better?
Perfect.
Yeah. Okay, so today I'm very excited to introduce Professor Molly Schumer, whose lab at Stanford University works on speciation, a lot of questions related to speciation, using natural hybridization events to understand like what barriers act to, I guess, reduce hybridization and keep species distinct, how these barriers evolve, and also like what are the outcomes of hybridization when it does occur. To do this, she primarily studies these xiphophorus fish species, which naturally hybridize. And in addition to like being like a pretty charismatic looking species with these elaborate ornaments, they also like show a large amount of like introgressed genetic variation in their genomes, which makes them like a really good system for studying like what sorry, using the words. I guess what the outcomes of hybridization are and what the impacts of that what are the functional impacts of that introgressed genetic variation. So please join me in welcoming Professor Molly Schumer.
All right.
I'm looking forward to this talk.
Thanks for the invitation and the introduction, Aiden. Yeah, so I'm excited to tell you about our lab's work on hybridization today. So from early in our history, evolutionary biologists have thought of speciation as this branching process, where ancestral lineage leads to daughter lineages that are reproductively isolated from each other. But even Darwin realized this was sort of an oversimplification of the process of divergence between lineages. But it's been in recent years, as we've been sequencing genomes from lots and lots of species across the tree of life, that I think we've come to a pretty clear understanding of how much of an oversimplification this was. And if we were to redraw this classic figure in evolutionary biology today, we might represent it more like a web with gene flow occurring before, during, and after divergence between lineages. And this is certainly true in the group that I study, the swordtail fish, which are a group of freshwater fish found mainly in Mexico and a little further south in Central America. And as Aiden mentioned, they're a really amazing model system for a bunch of reasons. One is these sexually dimorphic traits that have fascinated evolutionary biologists for a long time. They also have interesting ecological and behavioral adaptations. But what really drew me to this system was the potential for them as a model system to study hybridization and its impacts both in the distant past and in actively hybridizing populations. So my first PhD project was working with another graduate student trying to reconstruct the phylogenetic relationships between these species. And this was a phylogeny that had really had a lot of controversy, a lot of species moving around with handfuls of nuclear and mitochondrial markers. And so we used an RNA-seq-based approach for all of these species, and we built 10,000 gene trees and tried to infer the species tree using those relationships. And what we found is sort of consistent with the observation that these species can all hybridize in lab conditions, and some of them hybridize naturally. That is that even though we were able to infer the likely sort of branching history of this group, we also saw evidence on the phylogeny, and it's shown here with these swatches, of really pervasive genetic exchange between species. And this includes both very deep in the phylogeny, as well as between sort of contemporary species. And we actually know of many natural hybrid zones between some of these species, which I'll talk about today. So the fact that so many of the modern species in this group, and we now realize in many species across the tree of life, have substantial genomic contributions from hybridization, really changed my my thinking about how important hybridization was as an evolutionary process, and motivated me to really want to understand the mechanisms that are occurring when you have hybridization between species. So when we think about hybridization, there are a lot of different sources of potential selection on hybrids that might over long time periods kind of shape the dynamics of gene flow between species. And what I'll mainly talk about today is interactions between the genomes of the two parental species and how those might impact hybridization, as well as how they might arise and evolve. And I'll just tell you a tiny bit at the end about things that we're thinking about in terms of the way that ecological adaptation within species might impact hybrid fitness when parental species begin to hybridize again. And I won't have time to tell you about our work trying to understand the ways in which sexual selection, which is extremely important in this species group, may impact genetic exchange and the fitness of hybrids. But there are several ongoing projects in the lab led by two fantastic postdocs trying to evaluate this exact question. So the system that I'll talk about today mainly is a sort of major system in our lab, which focuses on active hybridization between two species of Xiphophorus that are sister species, Xiphophorus malinche and Xiphophorus birchmani. Malinche is a high elevation species. It lives in pretty cold temperatures. And birchmani is a lowland species that's adapted to much warmer and lower oxygen conditions in the warm sort of lake-like waters. And these two species hybridize at intermediate elevations. And as I mentioned, they are ecologically divergent. As you can see from the pictures, they're quite morphologically divergent, especially in particular sexually selected traits. And across the entire genome, they have about 0.5% per base pair sequence divergence. But what really attracted me to the system before I knew much about it was the structure of the hybrid zones. This is because these two species are distributed in river systems in the Sierra Madre Oriental of Mexico that, based on their distribution, form replicated hybrid populations. So basically, the parental species populations are fairly connected to one another at the headwaters and in the lowlands, but they each independently feed into these different river systems. And there are actually 7 river systems in which these species hybridize, but we focused our long-term sampling on 3 populations where we can easily collect many hundreds of individuals per year. And this gives us, a pretty powerful genomic database to ask about the impacts of hybridization across these independently formed hybrid populations in different river systems. So I think to date, we have over 3,000 whole genome sequences from these hybrid populations, about 1,000 per population. And I'll just walk you through briefly the kind of data we collect from these populations that will show up throughout the talk, as well as some of the basic things we've learned about the history of these populations from this data. So basically, the approach that we use is to collect whole genome low coverage sequence data. So, but during my PhD, I was trying to develop these methods to make it sort of cheap and scalable and accurate to infer ancestry along the genome of natural hybrids. And so what we ended up doing is generating these reference panels for the parental species from many different populations. And then we identified ancestry informative sites that were actually fixed differences between the parental species, no matter what population you sampled the parentals from. And so we have sort of a catalog of those sites along the genome, about 1 million sites for these two species. What we do is for hybrids, we collect this 1x whole genome sequence data, and we basically count across the genome at each ancestry informative site the number of alleles for Malinche and for birchmani in that hybrid at that particular site. And then we apply a hidden Markov model to infer what chunks of the genome likely originated from the Melanoma parent or from the Birchmanite parent. So what I'm showing you here is chromosome 1 of one of our hybrids from the Tlatomaco population. What you see sort of with the shading here is in blue are regions we have high confidence are homozygous Birchmanite in their ancestry. In red are regions we have high confidence are homozygous Melanoma in ancestry. And these unshaded regions with lots of markers are regions where we have high confidence that they're heterozygous for their ancestry. And so we can basically, for a given individual, have information about a given region of the genome. And then we can also, from the same data, infer genome-wide ancestry and population-wide ancestry. And so we've also been able to use this data to think a little bit about the history of these three major populations we sample from. And in particular, we've been interested in kind of characterizing the onset of hybridization. So one of the pieces of information from our local ancestry data that was most useful here was what we call the length of ancestry tracks. Basically the intuition here is that when you form an F1 hybrid, you have a whole chromosome from the Birchmanite parent and a whole chromosome from the Melanoma parent. And so the length of the ancestry tract is the length of the whole chromosome. However, with each meiosis, you're going to have recombination events between those two ancestries that are going to break those tracts into smaller and smaller chunks. And so we can use information about the length of the ancestry tracts, as well as some other information like the admixture proportions and decay and LD information to make inferences about when these populations most likely form. And when we do that for three populations, we see that they actually are estimated to have formed quite recently in the last 100 generations or so. And this is quite interesting to me, given my background, because it suggested that hybrids might still be resolving genomic conflicts that exist between the parental species genomes. And so I know many people will be familiar with this idea that hybrids will experience genomic conflict, but just to walk people through this kind of theoretical cornerstone in evolutionary biology, this idea of Dobzhansky-Muller hybrid incompatibilities. And so the basic idea behind this model is that you have some ancestral population that becomes separated for some reason, perhaps a geographical barrier. And those lineages that it forms, shown here in blue and purple, begin to evolve independently, and new mutations will arise in those populations, and eventually some of those will fix. And the mutations that fix within the population need to either be neutral and fixed by genetic drift, or potentially they might be under natural selection and be favored. But whatever reason, they're either sort of neutral or adaptive within population. But what Muller and Dobzhansky realized is that if these populations come back together and hybridize, in a hybrid, you'll be introducing mutations into the same genetic background that have not previously interacted. And so in a subset of cases, you might have negative interactions between these mutations that will reduce the viability or fertility of hybrids. And so what we know from decades of crosses of many species groups is that there's broad support for this model. It appears that interactions between QTL derived from different ancestries do seem to underlie reduced hybrid viability and reduced hybrid fertility in the lab and potentially in the wild. But one of the things that has always kind of bothered me about this is that because of the fact that the phenotypes that these interactions cause in hybrids are reduced viability or reduced fertility, it's actually very difficult to sort of drill down to the underlying genes. And so I'm showing you here a review from 2010 that lists at that time all of the known genes involved in hybrid incompatibilities. Even though there's been quite a lot of work in this area, there are only a handful of new additions in terms of precise mapping to individual genes since this 2010 review. And one thing that I think is especially important to note is that only a handful of genes that have been identified as involved in hybrid incompatibilities are active in naturally hybridizing species. So there's been a pretty strong reliance on model organisms because of the high throughput needed to map these interactions and sort of narrow them down, and the exceptional molecular tools needed. But as a result, that means we know very little, except for some cases like the PRDM9 master recombination regulator in mus hybrids, about genes that actually impact reproductive isolation in naturally hybridizing species. And so one might sort of say, as people have said to me at points in the past, like, why do you care about this? Like, we know the process that's going on. Why do we need to know the individual genes? But my interest in this is because I feel that without an understanding of the genes that become involved in hybrid incompatibilities, we're not able to test key hypotheses about what drives the evolution of barriers between species, especially young species. And there's been a lot of really cool theoretical work in this area, suggesting that things like substitution rates, genetic conflict, genome instability are likely drivers from a theoretical perspective of the evolution of hybrid incompatibilities. And these really fundamental questions become very hard to test without examples in naturally hybridizing species. So what we've tried to do is use the Swordtail model and in particular, a combination of our naturally hybridizing populations and controlled crosses in the lab to try to identify more of these incompatibilities and ask which of these hypotheses appear to be supported in additional hybrid incompatibilities that we can map. So today I'm going to tell you a couple of vignettes about some cases that we've made a lot of progress on, two where we feel pretty confident that we understand the genetics really clearly, and one that's a project in progress that I'd love to hear people's thoughts on. Okay, so my first vignette is going to focus on a trait. So this is a picture of a Xiphophorus birchmani male. You'll notice that on his tail, he has this collection of you'll notice a spot, and the spot is called by a collection of cells called macromelanocytes, which are sort of relative to the melanocyte cells that you see in other vertebrates. And so this is a sexually selected trait in birchmani, one of many sexually selected traits in this species. We were aware of it, but not very interested in it until we started collecting from some new hybrid populations and started pulling out hybrids that looked really crazy. So just to give you a sense of what these hybrids look like, this is a male that we collected from one of our natural hybrid populations. And we weren't initially sure if this kind of really crazy melanocyte-based phenotype was derived from the spot that we see in birchmani. But we raised individuals in the lab and also did genetic crosses in the lab, and we're able to see that basically there's this progression from a normal spot, like you see in birchmani, to this really expanded spot. And tracking individuals in the lab over time, we were able to characterize this actually as a malignant melanoma, which ultimately metastasizes. It typically will form tumors behind the eye when it first metastasizes, but an earlier effect is loss of the fin. And so we see that it has a pretty strong effect on swimming ability before it starts metastasizing. So we thought this was a really good candidate for a hybrid incompatibility. This is a phenotype we don't see in the parent species. And we do see in hybrids that impacts viability. And so we wanted to see if we could use our hybrid populations to get at the genetics of this trait. So we used an admixture mapping approach combined with some other GWAS-based approaches that I'm happy to talk about, but won't focus on in the talk. And we basically analyzed the data in a couple of different ways. So we collected hundreds of hybrids from natural populations that varied in their phenotype. We collected individuals that had no spot, individuals that sort of had this normal birchmani-like spot, and then these individuals with these really expanded spots that actually move into the muscle in the body. We basically analyzed the data such that we could identify the genes that were associated with just the spot and distinguish those from genes that were associated with the expanding spot or the early melanoma. I'm just going to sort of dive into the results here. What I'm showing you here is a Manhattan plot for the melanoma phenotype that we see in natural hybrids. And one of the peaks is a peak that's associated with Birchmite ancestry on chromosome 21. And we actually know from our other analyses is the peak that is associated with the presence or absence of the spot. So in order for hybrids to have melanoma, basically they have the presence of the spot allele from Birchmite. But then we also see when we look when we phenotype focusing on melanoma presence or absence, that individuals that have melanoma are much more likely to have ancestry from Malinche at this region on chromosome 5. And it's a little difficult to visualize just from the Manhattan plot. So if we sort of show this in like a genotype combination space, what we see is that individuals that have melanoma, which I'm showing here as what we call invasion frequency, so movement of the melanoma or of the melanocyte pattern outside of the tail into the body. So in order to have that phenotype, individuals have a Malinche allele, at least one, on chromosome 5, and at least one allele from Birchmite at chromosome 21 at the QTL peak. And we see that individuals that don't have both of those allelic combinations have basically zero risk of melanoma in our data set. And quite interestingly, even just having one Birchmite allele at chromosome 5 seems to quite significantly lower the risk that individuals are going to develop melanoma. And so I don't have time to talk about all that sort of fine mapping and GWAS approaches we did, but we were basically over using our hybrid populations and using populations of the parental species, we're able to narrow down these initial admixture mapping peaks that contain tens of genes to 2 genes that we believe are interacting to cause melanoma in hybrids. And these are the gene CD97, which is derived the risk factor is derived from Malinche, and the gene XMark, which is actually only present in Birchmite and has been deleted in Malinche. And so this is a pretty clear functional difference between the species. In terms of understanding CD97, we saw that there are both non-synonymous substitutions in a binding domain in this protein, as well as a pretty significant shift in expression that appears to be driven by changes in cis. So Malinche has very high expression of this gene, and both artificial and natural hybrids seem to inherit that level of expression, whereas Birchmite, and especially individuals with XMark, have much lower expression of this gene. And so we actually think it's a combination of both the amino acid changes and the expression level of CD97 that causes problems when combined with XMark, because we now know, in addition to this expression data, which had been documented in the literature to be associated with metastasis in human cancers, we actually now have results from cell lines where we pop in the Malinche version of CD97, and this actually promotes melanocyte migration. And so we think it's potentially a combination of both the changes in expression and the amino acid changes that seem to make this kind of a dangerous combination when you put the Malinche CD97 with the Birchmite gene XMark. So this was really exciting, and we felt like we were kind of finally getting down to individual genes that we were confident are causing incompatibility in Swordtail hybrids. But the same year our paper came out, we actually were super excited about this other paper that came from another research group working on 2 other species of swordtails. What I'm showing you here are their results of hybrids between Xiphophorus maculatus and Xiphophorus helleri. And you can see that these individuals also get melanoma and also have this sort of tail loss phenotype, but it has a slightly different progression. And these actually are species that don't naturally hybridize. So because they didn't have access to the natural hybrid populations, it took them a long time to produce enough crosses to fine map this interaction. But they found that it was actually also involving the X-mark gene and another partner, actually a different one from what we identified, this RAB3D-like gene. And so this kind of in concert with our results was super exciting to us because it suggested that X-mark was involved in both cases of hybrid melanoma, but the interacting gene differed. That was particularly interesting to think about when we think about X-mark across the swordtail phylogeny. So data from our group and others has really shown that X-mark has a very spotty distribution along the phylogeny. It is present in some species and absent in a bunch of other species. And we know from Birchmai and Malinche that in Malinche, it's been deleted due to a chromosomal inversion that basically splits the gene and makes it non-functional. And so this is actually a structurally different loss from other cases that we and others have looked at. And so it seems like X-mark has been recurrently deleted along the phylogeny. And our hypothesis, after sort of mulling over our results and results from Manfred Schartel's group, who led this other work, is the idea that X-mark might, when it's deleted, might sort of relax selection on interacting partners. So that deletion event might sort of change constraint in that network. And so that led us to hypothesize that these other cases where we see deletions of X-mark, if we take those species and we cross them in the lab, we might also see hybrid melanomas. And so we took this species Xiphophorus cuchianus that has an independent deletion of X-mark from the ones that have been studied and crossed it to Xiphophorus xiphidium. And we actually only are starting to get F2s that are mature now from this cross, but they have a very strong phenotype of this whole body melanoma. And it's actually a phenotype that even starts to show up in the F1s. So we're really excited to map this sort of independent deletion of X-mark and try to understand what genes that's interacting with in this cross to generate hybrid melanoma, if that is indeed the genetic architecture. And sort of more broadly, stepping back, try to understand the role that these sort of hyper mutable, or I don't know if that's the right term, but these regions that seem to be really prone to deletion might be having on a genus-wide scale in reproductive isolation in this group. And whether thinking about X-mark might be a model for trying to understand how predictable the evolution of hybrid incompatibilities might be more generally. Okay, so that was the first project I wanted to tell you about. And so I'm hoping that, next time I talk to you, we'll have some results for our F2 cross from xiphidium and cuchianus. But I'd like to switch gears and tell you about another hybrid incompatibility that we've been trying to understand and that we've recently been able to make a lot of progress on. So this incompatibility was one that caused me a lot of problems. So as a PhD student, when I started working on the system, I would go to the field and I would see there are lots of hybrids and I could collect them and sequence them. But I was trying to make hybrids in the lab and that was not working. And eventually I found that the cross actually only works in one direction with Malinche moms and Birchmai dads. And I'll actually circle back to this directionality of the cross in the last vignette that I'll tell you about in a few minutes. But basically, we found that we couldn't make the cross in the other direction in the lab. And as a result, all of our F1 hybrids that we made ended up with Malinche mitochondria. But that was fine. We could take F1s and cross them to each other and produce big F2 mapping panels. And to date, we've produced about 1,500 F2s between these species, which has been a really powerful mapping data set for both incompatibilities and trying to map the genetic architecture of different traits. So when I was first getting this data back, sort of towards the end of my PhD, I was, checking to make sure it made sense. And, with F1 intercrosses, you expect on average 50% of individuals at a given site are going to have Malinche ancestry, 50% Birchmanite. And so if you look across this example of chromosome 1, averaging across these 1,000 individuals, you see that ancestry across the chromosome is mirroring what we should see, which is great. But what we also noticed is that some chromosomes did not look like that. And in particular, a couple of chromosomes had massive deviations towards the Malinche parent. So chromosome 13 in particular had the strongest deviation genome-wide. And chromosome 6 also had a pretty substantial deviation towards Malinche ancestry in our F2 hybrids. And what we found when we zoomed in on these regions and looked at ancestry is that individuals with homozygous Birchmanite ancestry were completely absent in this chromosome 13 region, and mostly absent in this chromosome 6 region. And so this, combined with a couple of other analyses that I don't have time to dive into, hinted to us that we might be looking at potentially a mito-nuclear incompatibility, some kind of interaction between the Malinche mitochondria and Birchmanite ancestry at these chromosome 6 and chromosome 13 spots in the genome. And so, these are huge regions. And so we wanted to try to leverage our hybrid populations to fine map these regions. But one problem we ran into immediately is when we looked at our sort of big population data sets where we have lots and lots of hybrids, we found that all three of these populations were fixed for their mitochondrial ancestry. So they either had fixed the Malinche mitochondria or the Birchmanite mitochondria. And so this actually hinted to us, again, that probably something interesting was going on with the mitochondria, because since these hybrid populations are young, there aren't that many loci that are fixed in the hybrid populations themselves. And so we started sort of doing clinal sampling along these rivers and trying to find a population that wasn't fixed for mitochondrial ancestry. And so we identified one population where we found both mitochondria, the Birchmanite and the Malinche mitochondria. And what we did is we collected a ton of hybrids. I think we currently have over 600 from that hybrid population. And we tried to do admixture mapping using the mitochondria as the phenotype. So basically, we marched along the nuclear genome and asked, does ancestry at this nuclear locus correlate with the kind of mitochondria this individual has? And so I'm going to pop up the results for that. So we identify two very strong regions and one close to genome-wide significant region in this analysis from admixture mapping. And we, normally might sort of ignore a peak like this when we have peaks like this. But since this chromosome 6 region was already identified in our cross data in the lab, we did want to look at it. And so you'll notice chromosome 6 and chromosome 13, I've been mentioning those we see in our cross data. This third peak on chromosome 15, we did not see in our cross data, and I'll get to that in a moment. But basically, we actually, in this peak in particular, had exceptional resolution because this is a high recombination rate region in the genome, it turns out. And there are only 2 genes in this region. And one is the mitochondrially interacting nuclear protein NDUFS5. And so observing that, we then looked at the 10 or so genes in these other regions and noticed that this chromosome 6 peak has this gene NDUFA13, and the chromosome 15 peak has this gene NDUFA-F4. All of these genes have the same kind of name because they form a protein complex in the mitochondrial electron transport chain. In particular, NDUFS5 and NDUFA13 fall right together in this inner membrane space of this large protein complex that is essential for the function of the mitochondria. And NDFA4 is actually also involved in this complex. So sort of telling you a little more about the genetics that we're seeing here. If we look at NDFA13, and we kind of break down the genotype combinations, what we see is that individuals with the Malinche mitochondria and homozygous Birchmai ancestry at NDFA13 are missing from our data set. We expect to see a lot of these, and we see none. This is consistent with our mapping results, where only less than 1% of individuals in our cross in the lab had this genotype combination. Moving to this chromosome 13 region, again, we see individuals with a Malinche mitochondria and homozygous Birchmai ancestry now at this gene NDFS5 are absent from our data. Again, in our 1000 F2s, we never saw this genotype combination. What's interesting is that because this is a hybrid population with both Birchmai and Malinche mitochondria, we can look for interactions with the Birchmai mitochondria, which we could not do in the lab because we've never gotten that cross to work. And what we can see is that there appears to also be selection on interactions between the Birchmai mitochondria and being homozygous Malinche at NDFS5. And so we see that there's sort of reciprocal selection on this interaction, which is actually something that's to totally be very nerdy about, is something that's not really expected from the theoretical literature and is a cool thing to be able to see in empirical data. I'm gonna sort of drop off and not talk about chromosome 15 and NDFAF4 for the rest of the talk. And the main reason for that is we haven't yet been able to study it very well in the lab. Again, because we haven't been successful at making artificial hybrids with the Birchmai mitochondria in the lab. So for the rest of the talk, I'm going to focus on what we believe to be a sort of complicated mito-nuclear interaction. We're having the Malinche mitochondria and homozygous Birchmai ancestry at NDFS5 or NDFA13 is either completely lethal or largely lethal in hybrids. We felt really excited about how far we were able to push the genetics in understanding this hybrid incompatibility. But I'm always sort of curious about how far we can go with this very non-model system. And so one thing that Ben Moran, a grad student in the lab who's been leading this project, did was try to see what we could learn from the protein structure of mitochondrial complex 1, which has been intensively studied because it's important in human disease and basic cell function. And what we found, interestingly, was the predicted protein structure of mitochondrial complex 1 using just the protein sequences from Birchmai and Malinche was essentially identical to all other eukaryote complex 1 predicted structures and known structures using cryo-EM to identify those. And so he was able to sort of visualize then where in this full protein complex 1, which has about 40 proteins, the mitochondrial genes were. And so those are shown here, as well as where the interacting nuclear genes with the mitochondrial proteins are showing up in the structure. And what was really cool is basically he sees that NDFS5 and NDFA13 are right next to each other in physical contact in mitochondrial complex 1. And they're both in contact with a third gene, a mitochondrially encoded gene, ND6. And so this gives us hints that ND6 is probably the gene in the mitochondria that's involved in this incompatibility. And what was cooler to us was that with the protein modeling, he could actually visualize where the substitutions between species fell on these proteins. And he's highlighted these here in spheres. And what you can see is that both NDFA13 and NDFS5 have substitutions that differ between species that are predicted to be in protein-protein contact with substitutions in ND6. And so this is a hint at a potential mechanistic explanation for what's going on here. And so we've made a little bit of progress kind of pushing this further from modeling. One challenge is that we don't have viable hybrids with the incompatibility. They do not survive. I'll show you a little data on that in a moment. So it was kind of hard to look in particular at the function of complex 1 in individuals actually impacted by the incompatibility. But what we decided to do was to look at F1 hybrids, where individuals are perfectly fine. They survive to adulthood, they reproduce, but we reasoned that they should have some reduction in complex 1 function if the incompatibility is indeed acting right here. And that's exactly what we see when we measure that basically the enzymatic activity of mitochondrial complex 1 in Birchmai, Malinche, and hybrids. In F1s, we do see that it's significantly depressed relative to the parental species. And what's a little more interesting to me in sort of thinking about what's going on at an organismal level is that individuals, F1 hybrids, do sort of get to a parental-like level of complex 1 activity, but it takes them a super long time. And so I think this could be a hint that F1s are compensating in some way, given that they do have some functional allelic combinations that they can sort of make functional complex 1 eventually, but they need some time to do it. Okay, so I'm gonna just tell you one more thing about this particular incompatibility, and that's our trying to think about the architecture of the incompatibility. So as many people will know from theory, there's been this prediction that complex incompatibilities, those involving more than 2 genes, 3 or more genes, should be common because they're easier to evolve in sort of finding paths through mutation space to these incompatible gene combinations. And so I was interested in trying to think about whether this is what we would call a true throughway incompatibility, or whether it's more likely to be kind of a combination of two-way incompatibilities. And this may seem very much in the weeds, but I think it has important theoretical implications for how these barriers evolve. And we don't have a full answer to this, though we're working on it. But we have some hints that they are actually sort of separable incompatibilities in their function. So in particular, what we see is individuals that have mismatch at NDOFS5, which I'm showing you here in red, do not survive embryonic development. They basically start embryonic development, but they never finish it. They stall out at sort of midway through embryonic development. We do not see this with individuals that have mismatch at the other gene. And so I think this sort of, those individuals die sometime between birth and juvenile life stage. So I think that sort of temporal separation in when there's lethality suggests that this isn't a true complex incompatibility, but it's actually sort of two different incompatibilities interacting with mitochondrial complex 1. And to me, that's exciting because it kind of, again, points to this hotspot hypothesis that I've been lately obsessed with, that maybe certain pathways or certain interactions are kind of fragile, either due to their evolutionary or molecular properties, and they may be especially prone to breakdown in hybrids and cause these kinds of problems. Okay, so that was the second project I wanted to tell you about. For the last couple of minutes, and this will be a very short vignette because it's an ongoing project, I wanted to tell you, sort of return to this observation that very much frustrated me during my PhD, that we could really only make crosses in one direction. And I think that there's a really interesting interaction here with the ecological environment that we're just beginning to explore. So I told you earlier that we were only successful in making crosses between Malinche moms and Birchmai dads. And when we try to make the cross in the other direction with Malinche dads and Birchmai moms, all of our broods so far, with over 900 offspring born from those broods, are born early. So they're prematurely born. This is a picture of one of those broods. They still have an egg yolk. Their heart is on the outside of the yolk. And this is very, very fatal. We've tried to culture these guys in vitro when they're born, but lethality is well over 98% in the first week. And we've only ever had 2 survive to adulthood. So this is sort of an extreme embryonic incompatibility, and we've long wanted to know what the cause of it is. I hadn't really made much progress in it or even thinking about it until a graduate student in the lab noticed that of all the babies that are born in the lab, oh, let me back up. Sorry. Swordtails are like mammals. They give birth to live young. The babies cannot survive outside the mom, and when they're prematurely born, that it leads to high mortality. It is poorly understood why this is the case, because it's thought that what's going on in Xiphophorus is they just keep the eggs inside the mom, and there's no real sort of maternal resource transfer during pregnancy. However, one of the graduate students in the lab noticed that of the 5 species we have in lab, the babies really differ in size. And in particular, Malinche have extremely large babies, about 30% larger than any other species and 50% larger than some of its close relatives. And so this caused us to wonder if there might be some kind of size incompatibility in this cross, like has been seen in certain mammalian crosses. And so we, hadn't really looked at this because the offspring are born so prematurely in one of the cross directions. But we started sort of looking back at earlier stages of embryonic development. And so we could see that this size difference between Malinche and Birchmai arose earlier in embryonic development. This is developmental stage 30. So that's sort of consistent with what we see at birth. And quite strikingly, F1 hybrids in either direction are extraordinarily large. We're still kind of early in trying to understand what's going on, but our hypothesis is that, even though the F1 hybrids are large when Malinche is the mom, they're used to the hybrids, they're used to their normal offspring being larger. And perhaps this combination of extraordinarily large offspring in moms that are not used to their offspring being so big is what causes this premature birth phenotype. We've been trying to also understand what developmental stage this is happening at, and we're actually doing a pretty large crossing experiment to track individuals through development now. But we do have data for the parental species. And what we see is that Birchmai Malinche eggs start off about the same size. So it's not the Malinche eggs are larger to begin with. But Birchmai eggs lose weight during embryonic development. Malinche eggs do not, and they're born about the same size as they are at fertilization. And another thing that we've noticed in characterizing this and dissecting these fish is that Malinche has highly vascularized tissue around the embryos, whereas Birchmai does not. So you can see all these little blood vessels that sort of surround each individual embryo. This we see in Malinche, but not in Birchmai. So our hypothesis is that Malinche moms are actually transferring nutrients to their offspring during development, which is why their offspring don't lose weight over the course of development, as opposed to Birchmai offspring, where they're using up those maternally provisioned resources and eventually losing weight before birth. And we're just starting to get in to try to understand the genetics of this, but Cheyenne's done some RNA-seq experiments sequencing this maternal tissue and seeing that in Malinche, there's upregulation of genes that are associated with placentation in other fish species, as well as in mammals. So again, this is really early days, but we think this is an exciting divergence between these species that may be responsible for this premature birth phenotype. And this is finally sort of something that kind of moves us out of the gene by gene interaction level and thinking about the ecological differences between these species. In particular, as I mentioned earlier, Malinche lives in extraordinarily cold environments for this genus. So it's the highest elevation species in the whole genus. The waters it lives in get down to 7 degrees. And as a result, there aren't that many insect larvae or as much algal growth in their environments compared to the low elevation species, which is sort of the ancestral habitat. And there's been work in guppies that has shown that artificial selection, where there's resource deprivation, actually can trigger this evolution of larger offspring size. And so our basic working hypothesis is differences in the sort of resource availability in the ecological environment may drive some of these evolved differences in offspring size between Malinche and birchmani, and potentially have these secondary effects on hybrid fry size that may be responsible for this spontaneous abortion phenotype we see in one cross direction. Okay, so I'm going to wrap up there and just say that I've told you a lot about genetic incompatibilities. There's a lot more that we hope to learn. And we're also hoping to push this into sort of a more global understanding of why certain phenotypes evolve and what their impacts on hybrids are, as well as how the behavioral environment is important in hybrid populations as well. And I've talked this whole time about swordtails, but these are questions that are relevant to other species across the tree of life. And I think the coming years, as we've appreciated the importance of hybridization, we'll learn a lot more about swordtails and other species groups. I'd like to acknowledge the people who worked on these projects from my lab, as well as some awesome collaborators and funding sources, and happy to take any questions.
Hey, Molly, we just clapped, but you probably couldn't hear it. That was great. Do you want to take questions, and I'll pass them on if she can't hear? Sure. Yep, turn that on. And folks on Zoom, you can type it into chat or something, or raise your hand. Are there questions? And Molly, if you can't hear the question, let me know and I'll repeat it.
Okay.
Hi, Molly. Great talk. I was interested in the timing of hybridization between these two species. I'm not sure what the generation length of these fish are, but 100 generations seems pretty recent. I'm wondering if you could talk at all about, like, I don't know what you think might be facilitating the hybridization, or why it's kind of happening relatively all of a sudden.
Yeah, for sure. So we do think this is an anthropogenically linked hybridization event. There are a couple of rivers where that are extremely hard to get to, where we don't find hybrids. And what we see is that the rivers where hybridization is most frequent are the most impacted. We have a collaboration where we've been measuring water chemistry. And there seems to be a pretty clear link between sort of degradation of the water environment from agricultural runoff and the onset of hybridization. And Gil Rosenthal's group has shown in the lab that if you add, some of these common contaminants to the water, you basically sort of block olfactory communication. So that's the mechanism that we think is important in the recent hybridization event.
Okay, interesting. Thank you.
Oh, we got a question on Zoom. Emily asks, the increased embryo size in hybrids reminds me of mother-child conflict. Do you see any changes in health of mothers carrying hybrid offspring?
Yeah, so great question. So in Malinche moms, we do not, but the birchmani moms, when they're carrying hybrid offspring, in addition to the two phenotypes we see are this premature birth, we also see maternal lethality. So sometimes those birds seem to kill the mom.
Hey, Molly. It's Bill Prescott. How's it going? Great talk. So I have a question about the X mark results. I wanted to see if you might go out on a limb a little bit, which is that you can imagine that really what's going on with the X mark is that when it's being controlled developmental genetically, then it actually is functioning, but when it's released from that restrictive molecular genetic environment, it becomes malignant and cancerous. So to go out on a limb, I'm wondering if you think maybe that could be kind of a general place we might expect to see Jansky-Muller incompatibilities, is where one interacting partner needs to be controlled, and there's multiple different ways to control it. And then in hybrids, basically, that control is released. So anyway, I just wanted to see if you go out on a limb a little bit and think about.
Yeah, I think I got your full question, but it was coming in and out a little bit, so let me know if I don't answer it. So yeah, I do think there's something like that's important to be learned about the structure of genetic networks and hybrid incompatibilities. And I think this is another reason, like, we've got to map more of them, because I do think there's, reasons from first principles to believe that, like dense genes that are sort of dense and sort of maybe at the center of genetic networks, might be more fragile to sort of changes in any of their interacting partners and sort of drift in function. So I think that's something I would really love to know more generally besides XMark.
Hi, that was an awesome talk. I was wondering, so you guys said that you were not able to make any like F1 hybrids with, I'm gonna garble the fish species name, but one of the moms was not able to hybridize with the other, but you guys found hybrids out like in the field, and I was wondering if you guys had any intuition as to why that was able to happen in nature and not the lab.
Yeah, so our thinking is basically that this phenotype must, segregate at some point. Like, if there's this kind of hard block on the F1s where they're not, we can't get those viable hybrids, but we do see hybrid individuals with birch-knight ancestry in the wild. If you have, recombination in the natural hybrids and then back crossing with birch-knight, are sort of only, and we don't have really direct evidence of this, but our interpretation is that there must be some point at which they are viable in that cross.
I think given the time, we should probably thank Molly again. But are we hanging out afterwards? When's that? Okay, so Molly, there's a general hangout with you at 4, is that right?
Yes.
Okay, cool. How do people find out about that? I guess if you want to be in on that, ask David.